Hi, I'm Stephen Bodine. I am a graduate student in the Muir Lab. And one of the things that makes humans so incredibly interesting as a species is our ability to communicate. This communication ability enables us to respond to threats, respond to changing environments in an incredibly dynamic and fast way that optimizes our biological fitness, which means our ability to reproduce and pass on our genes. Now, this isn't unique to humans. Humans aren't even the only mammals that can do this. Pods of dolphins or packs of wolves display similar communication behaviors that coordinate their group behavior. However, it might surprise you that lower organisms can exhibit similar types of behavior. These patterns of behavior are observed in social insects like bees and ants that communicate with scent and physical movement like dances. But even the seemingly simplest organisms, bacteria, also possess these communication systems. However, bacterial communication is a chemical communication system. What these bacteria do is they secrete a small chemical signaling molecule into their environment so they can communicate with other individual bacteria localized to where they live. This communication system relies on the concentration of that molecule that's put into the environment. The bacteria can dynamically sense increasing and decreasing concentrations of that molecule, of that communication signal, and then respond in such a way that they coordinate all of their behavior for optimal biological fitness. And this entire process is called quorum sensing. Again, trying to monitor what the local population density is. In the Muir Lab, one of our major project areas is to study the process of quorum sensing in Staphylococcus aureus, which is a bacteria they might be familiar with due to its propensity to pick up antibiotic resistance. So you hear it on the news, most commonly MRSA, or methicillin-resistant Staph aureus. So what we do here in the Muir Lab is we try and study this system by reducing it to its base components, taking it apart and recapitulating each piece of what makes up the entire process in the test tube. So this kind of work requires precise molecular access to each of the constituent components of this system. Now, as in any biological process, it's a combination of small molecules, so chemicals, interacting with proteinaceous proteins in whatever small molecular compartment or environment that these systems operate. So what we have to do is be able to do both: synthesize the small molecules that participate in this system and express and purify the proteins that are interacting with all those small molecules in this biological pathway. Now, the hardest part about this is not the chemical synthesis. We have a very easy way to get to these simple biomolecules that these processes are working on. The problem is the fact that all of this process, this entire system, occurs in the bacterial membrane, meaning that we have to access membrane proteins, which generally don't function properly when you just put them into a test tube. You need to recapitulate their membrane environment in some type of model system to be able to get them to work properly so that we can study the system in the testing. So what I'd like to do now is show you one of the ways that we are able to purify and assemble these macromolecular assemblies of lipid and protein to get these proteins to act properly in a facsimile of their native environment. So this machine is a fast protein liquid chromatography, what we refer to as FPLC instrument, which we use to do a technique called size exclusion chromatography. Now, essentially what we're doing is when we try and assemble these lipid protein macromolecular assemblies, essentially little sandwiches of membrane protein within lipid, we need to purify the fully assembled species away from the materials that we use to make these systems. And the way we do that is we separate them by size. So we put them onto a column called a gel filtration column that has tiny pores in it. And if you come closer and look at the system, right now it's showing an example protein coming down through it, colored red. What happens is the sample is injected into the top of the column, and as it passes through the gel filtration column, it accesses tiny pores in between each of the little beads present on that column. The bigger the species is, the fewer pores it can go into because it's too big to access some of the tiny spaces between beads. So it comes off the column quickly. Whereas small species, meaning the kinds of things that we use to assemble these protein complexes, can access more of the space in between the bead matrix, and therefore it will take a lot longer to pass through the column, thereby allowing us to separate our fully purified assembled species from any other impurities or contaminants within the system. So thanks for listening, and if you want to hear more about Quorum Sensing and Staph aureus, feel free to reach out to any of us here at the Muir Lab. We'd be happy to chat.